at home, in your workshop, you have all the wood and tools you need to make the dresser. You follow the instructions and make a good-looking dresser effectively, translating the written words into a physical object that you can use in your home. This analogy has set the stage for us to understand the central dogma on a molecular level. The library represents the nucleus, and your workshop at home represents the cytoplasm. All the books in the library represent the DNA, the genetic material of cell stored in chromosome. Nucleus is usually a round part of most cells that is enclosed in a double membrane. It controls the activities of the cell and regulates the activities and contains chromosomes. It takes 10% of the cell volume. Cytoplasm is a thick solution that fills each cell and is enclosed by the cell membrane. It is mainly composed of water, salts, and proteins. It provides a medium for the organelles to remain suspended and protects the genetic material of a cell and also cellular organelles from damage caused due to movement and collision with other cells. DNA, that is deoxyribonucleic acid, a self-replicating material which is present in all living organisms as the main constituent of chromosomes. It is the carrier of genetic information. The instructions for the wooden dresser you chose represents a single gene. As you may know, each gene contains the instructions needed to make a single type of protein. The finished dresser then represents the protein product. The wood used to make the dresser represents amino acids, which are the building blocks of proteins. Amino acids are organic compounds that combine to form protein. Amino acids and protein are the building blocks of life. Gene is the basic physical and functional unit of heredity. They are made up of DNA. And what about the notebook? It represents RNA, which is a small molecule that can move out of the nucleus into the cytoplasm, where proteins are made. Simply put, the central dogma states that DNA leads to RNA, in turn leads to protein. RNA is ribonucleic acid present in all living cells. Its principal role is to act as a messenger, carrying instructions from DNA for controlling the synthesis of proteins. Complex proteins are made up of various protein molecules, which are linked by non-covalent bonds. Amino acids are the basic units of the protein. The synthesis of proteins occurs according to the central dogma. The central dogma explains how genetic information flows from DNA to RNA to make a functional protein. It suggests that DNA contains all the information required to synthesize a protein, and the RNA acts as a messenger which carries this information to ribosomes. Ribosomes is a type of cell organelle. Non-covalent bond means they do not share electrons. Transcription results in the formation of messenger RNA using DNA as a template. This process takes place in the nucleus. You must be aware that DNA is made up of 2 strands. But in this process, only one strand of DNA is required, and this one strand is used as the template for the synthesis of messenger RNA. An enzyme, RNA polymerase, helps in this process. It helps in copying the information from DNA strand to messenger RNA. Once this process is complete, the RNA polymerase dissociates from the strand, and the newly synthesized messenger RNA is released from the nucleus. Like DNA, RNA is also made up of nucleotides, but there is a difference. RNA contains the nucleotide uracil instead of thymine, which is present in DNA. This uracil pairs with adenine just like thymine. Once the process of transcription is complete, the newly synthesized messenger RNA is released from the nucleus into the cytoplasm for translation on ribosomes, producing a polypeptide.